Ok. Ok. Du tout, ouais. Ouais. You decide.
So, hello everyone. So today we have the pleasure to welcome Olivier RIVOIRE. So he's working at ESPCI. So he's a physicist, but interested in various topics related to evolution, biology. And so today he will talk about minimal physical models with evolutionary features. So, thank you.
Okay, well, thank you very much, Jorge, for the invitation. So yes, I would like to talk about minimal models, so very simple models, but for a problem that itself is not so simple, which is the problem of, in general, what is life. And as you may know, Schrödinger wrote a short book in the 1940s with this title, where he was inviting physicists to look into biology for new laws of physics. We are more than 80 years later. There have been a lot of progress in biology, but if we look at it from the standpoint of theoretical physics, I mean, there has not been so much progress. We don't really have even like a simple Ising model that people will agree is a good model for representing life, for understanding what kind of state of matter it is, or what kind of class of dynamical system it represents. Now, it's not an issue for biologists who, in general, do not even bother having a definition of life. There is just one exception, which are biologists interested in exobiology, okay, so trying to look for life outside of Earth, and they need to know what they are looking for. And so this is in particular a concrete problem for the NASA. And a few years ago, they gathered the best specialists they could find, okay, to come up with a definition of life. And this is a definition that they proposed. Life is a self-sustaining chemical system capable of Darwinian evolution. Okay? And okay, it's a definition, and we can always argue about definitions, but it's a relatively well posed definition, and we can start to ask, okay, what will be a minimal physical model that will have all these features? Okay, and this is the question that I'm going to address here. And for that, we need first to specify a bit what is meant by Darwinian evolution. And this is well understood since Darwin, obviously. And so what's Darwinian evolution? It's the idea that once you have a few principles that are met, namely you have a population with entities that can reproduce, these entities have different properties, different traits. Depending on these properties, they will reproduce more or less. They will transmit those properties, at least in part, to their offspring. And then there is a diversity of different entities. And from this principle, then you have evolution by natural selections, and you have a number of different properties that we find in general in biological populations. So this is the way in which evolution, Darwinian evolution, is usually presented. Now, if you really want to get deeper and you ask, okay, so what's really meant by reproduction, heredity, diversity? If you look into biology, the way in which, for instance, heredity works, involving, for instance, DNA replication, it's very complicated, okay? And there are a lot of molecular mechanisms, enzymes, and so on, that are involved in the mechanisms behind these properties. And the best way we have to explain those mechanisms will be, again, in terms of Darwinian evolution. Okay, so there is a bit of a circular reasoning in the sense that the basic principle of Darwinian evolution, the way in which they are implemented in biological systems, we cannot explain them without referring to Darwinian evolution. So the question is, okay, what can we think of something more fundamental, more physical, okay, from which we can start this recursion? And there is, in fact, a relatively clear agreement on what should be on the top. Namely, the idea would be to start from objects that can self-replicate. And if you think of it in a chemical context, it means autocatalytic systems, so molecules or set of molecules that will promote the formation of more molecules of the same type. And there are different ideas and theories, particularly in the context of the origin of life. So there is a school that is called genetics first, who think that the first replicators were a molecule like RNA, so a complex molecule that is able to make copies of itself. Another view is called metabolism first, where instead of thinking of a complex molecule, people think of a collection of small molecules that are interacting in a network of reaction. And this is called metabolism first idea. There are many other proposals. So, for instance, Freeman Dyson, who was one of the few physicists who got interested into this question, proposed that, in fact, the two things started independently and fused later on. There are also proposals where it's neither genetics first or metabolism first, but something else. And most of it comes from the view that the way in which we think of living organisms in terms of chemical reaction is the coupling between three different systems. One that is really metabolism, another one that is more template replication, genetics, and then the fact that you have a membrane. Okay? And each of these systems is itself or can be autocatalytic, and the question that these people ask is, okay, which one started first, and so on. Now, from the point of view of physics, what we can ask is, okay, how can we conceive of a very simple physical system that has this ability to be autocatalytic? And in fact, it doesn't need to be very complicated. So this is an example that we analyzed with the students a few years ago, where you just have, you can imagine it's just two Brownian particles that can form a dimer. And by interacting with monomers through a cycle, that is a catalytic cycle, it will promote the formation of another dimer. Okay? So it's a very simple form of autocatalysis, and we can work out all the conditions that you need for this to occur, for instance, with exponential amplification. So this is clear. What's really less clear is how do we go from self-reproduction or self-replication to dynamical behavior, where we see a lot of variation that is inherited and something that really looks like Darwinian evolution with all these different features. And at the moment, there is no proposal where really starting from a simple network of reactions that display autocatalysis, we can achieve all these properties. So what I would like to present today is a model that we developed with Guy Bonin, where we take a different view, and instead of starting from self-replication, you will see that we start from other principles and we'll get self-reproduction or self-replication to occur as an emergent feature of the dynamics. Okay? And instead, the principle from which we start are these three principles. So there will be an open system with influx and dilution, a set of chemical reactions with a key feature, which is the fact that this reaction can display multi-stability. And then there will be an important role played by space. And we will see that from this principle, we'll get all of these different properties. Actually, not exactly in the order where we will first get this list and then this one, but in a slightly different order. And so in the rest of the talk, I'm going to go over each of these items and explain you what they mean and how they are related to each other. So I will start with the first point, okay, what we mean by an open system. Okay, and maybe, yeah, as you will see, I mean, what's also interesting is what is not in this list. So I was explaining before that autocatalysis was thought to be a key element. You will see that we don't need it here to get these different elements. There will be also no compartments involved, not even large molecules, and you will see also no clear distinction between genetics and metabolism. But let me start with explaining the first principle from which we start. So we imagine that we have a physical system that corresponds in fact to an open reactor, okay, where we have some particles, some building blocks that are continuously injected, and then also continuous dilution so that we keep a fixed volume of the reactor. And the particles that we are going to inject will be of a few different types. You can think of them as molecules or colloids. What's going to be important is just the fact that you have Brownian motion and they will be able to interact with each other. And I'm going to start with a situation where this reactor is well mixed. Okay, so there will be at any given time only one concentration for each type of elements. Okay, so this is the first thing. Then what we want is in this reaction, in this reactor, to have chemical reactions displaying multi-stability. And so one way to implement it is first to imagine that in input, we have these dimers, and we also have this catalyst. Okay, so there are different particles with different interactions, and they are such that this particle is promoting the dissociation of the dimer. Okay, and we can, in fact, I'm not going to explain details, but there is a way in which you can really design the geometry and the interaction of very simple particles so that you will have this kind of behavior where by itself this is quite stable, but when it interacts with these other elements, is going to facilitate the dissociation. Okay, so this is the first element. There is production of some particles A1. Then we imagine that, okay, in this reactor, actually the volume is finite. Okay, so there will be fluctuation from the finite number of particles. So if we look at the evolution in time of the number of the concentration of A1 particles, so there is a term of production, there is a constant dilution, noise, and as a result, it fluctuates around some value. Now, the idea is to have bistability, and for that we'll have more than one reaction. And the basic idea to get first bistability is to imagine that you have two reactions that are coupled in the sense that the product of one is inhibiting the second, and reciprocally, the product of the second one is inhibiting the first one. Okay, and later I'm going to represent it in this way. And the idea is that with this kind of inhibition, if we start with a high concentration of A1, this will turn off this reaction, but still more A1 will be produced, and so we'll have a state of A1. And on the opposite, if we start with more A2, then we'll get a state of A2. And in practice, if we want to implement it with simple physics, the idea is, so we have this production of A1, which is what I was showing before. And then we imagine that, okay, we have the same for the production of A2, but then A2 can bind to C1, and by binding to C1, it prevents this reaction from occurring. Okay, so this is the idea, and you have the symmetric reaction for 2 instead of 1. It turns out that this is not quite sufficient to get multi-stability, which is something that is well known in biology. But just adding one more ingredient is sufficient. This ingredient, I mean, you don't need to care too much about it, but it's called ultrasensitivity. It's the idea that, in fact, you need to have sufficiently high concentration of A2 before it will have an effect, and this is done by having it interact with another type of molecules. So at the end, what we have in this case is just two coupled equation corresponding to this production of A1 and A2. So you have the production that possibly is inhibited by the other molecule, dilution noise, and then the symmetric case. And in this case, you can get dynamics with bistability, with switches between one state and the other. And these two states correspond to either A1 high concentration, A2 low, or the opposite. And this is represented in this way, where you have the two reactions, and black means high, and white means low concentration. Okay, so that's bistability. Then next, we are going to generalize it to have more reaction and get more states. So first, I can give a simple illustration with 4 reactions. And so this is represented by this graph. And so each node corresponds to one reaction, the production of AI. And then these edges represent reciprocal inhibition. Okay, so that A1, 1 is inhibiting 2, and 2 is inhibiting 1. And so if you have more reaction, you can also have more states. So in this particular case, you have 3 states, and it's not too difficult to enumerate them. What you need to have is every time you have an active reaction, which is represented in black, then none of the neighbors on the graph can be active because there is inhibition. And if you just enumerate all the ways in which you can color your graph so that there is no black neighbors to a black particle, then in this case you will find these 3 states. Now, if you simulate these dynamics, what you will observe as before, you will observe transition between different states. But the first interesting observation is that, there are some colors that are not displayed. But the first observation is, in fact, if you look carefully, you will see that there are only 2 states that are visited by the dynamics, which are these 2. And this one, where A2 is high, is actually almost never seen, even though in this case we started from a configuration in A2. Okay, so the system switches between the states, but the transition from one state to another is not uniform, and there are some transitions that are preferred. And so this is going to play a role in what follows. So let me explain a bit more where this is coming from. And we can understand it in a very simple 2-state system. So this is a case where you have, again, here 3 reactions. And you have 2 possible states, which are represented here. And what happens in this case is that you will transition more easily from this state to this state than the opposite. And if you look a bit at the representation of the dynamics, you can understand quite easily why it is the case, because to go from here to here, you need a fluctuation of the blue one that goes down. If you want to go the other way, you need a fluctuation of these two concentrations at the same time. Okay, so here you need the orange and the red to go down spontaneously at the same time, and this is obviously much less likely than the opposite. Okay, so this is the idea of why there is a bias. It can be formalized a bit more. We can introduce something that we call productivity, which is representing essentially the number of active particles, so the number of black dots in the graph. And what we can show is that this productivity, you can think of it as minus an energy, and we can show that there is a detailed balance relation where the relative rates of the transition from one state to another can be expressed in this way, where again this productivity, which is a number of black nodes or active reactions, is like an energy, and where the temperature is related to the stochastic noise that induces the transitions. So, okay, and so next we continue to generalize. So, and we are going to consider a large number of chemical reactions. What's going to be important is that we will have a graph of reciprocal inhibition between these reactions that is going to be sparse, meaning that each reaction is inhibiting a finite number of other reactions. And then, as before, it's reciprocal. So when one reaction, the product of one reaction inhibits another one, then reciprocally, the product of the other reaction inhibits the first one. And in this case, we are going to take, for illustration, a random graph of finite connectivity where every reaction inhibits exactly 3 other reactions. And so in this case, if you enumerate the number of possible stable states, you can show that they are exponential in the number of reactions. So in what follow, I'm going to use 50 reactions, and you can have 500,000 states of the system, okay? Where again, one state of the systems corresponds to a subset of the products that are at high concentration and the other that are at low concentration. Now the idea is that you have this system in a reactor, okay? You start maybe with nothing active, you will reach a state, and then you have fluctuation that lead you to access a different state. As I said before, the transitions are biased, okay? So you tend to go towards some states. So if you look at the dynamics over a long time scales, in fact, you will see that the number of active reactions, so this corresponds to the productivity that I was introducing before, is increasing slowly in average over time, okay? And it occurs over a very long time, okay? So this is a notion of directionality that is of interest when we think about evolution. Now this transition, when they occur, typically you have one product that was high concentration that becomes low concentration, so there is just a small change in the definition of the states. And if you look at the correlation between successive states, not only they are correlated, but they become more and more correlated as time goes, okay? And so this you may recognize is a signature of aging in disordered systems. And in fact, you can make a mapping between these models. So this is what I was also started to present with this analogy between productivity and energy. Okay, and so this system is formally what we may call a chemical glass, okay, where you have these different states that are analogous to the different configuration of a glassy system. Okay, and so this is why we have this long dynamics over long time scales, and we have this increasing correlations. So, okay, so what have we seen so far? So I introduced these 2 principles, and already we have a system that displays a lot of diversity. There is a notion of heredity in the sense that the successive states are correlated to each other. And then we also have directional dynamics over very long time scales. Now, next, we need to talk about reproduction. Okay, so previously I said, okay, in many models, people are really taking it as a starting point, okay, making a self-reproducing system, an autocatalytic system. Here, it's something that, okay, is not in the list of principles. This is something that is obtained as a result, and the critical ingredient to obtain it is to introduce space in the model. So what is meant by space is, instead of considering a well-mixed reactor, we are going to consider that diffusion is limited. Okay, so in this reactor, we keep injecting uniformly the building blocks all over space, but then it's not well mixed, meaning that you will get different concentration at different locations. So now if we come back to the simple 2-state system that I introduced before, and so if you put it in space, and here it's going to be a one-dimensional space, and here the reactor starts in the state, the the opposite. Okay, in the state 1, but what you will see that you get a transition at some point to the other state, and then this state expand over space and eventually invade the system. Okay, so there is a notion of reproduction in the sense of growth of one phase over the other. Okay, so far what we've seen, there is not really new physics. I mean, we are able to map the observation to previously known phenomena. So we had this system of coupled reactions. When it's well mixed, essentially it corresponds to a mean field glass, okay, because we are using a random graph. When we introduce space, I've introduced it in the context where we have just a few reactions, then we have this kind of pattern that is very much reminiscent of nucleation and growth. And so these two limits are two limits that are very well understood physically. But then where we want to go is here, where we have both a large number of types, and then we have a system that is expanding in space. And as far as I know, there is no general theory for what occurs here. And what I'm going to do is to show you that, okay, there are some interesting features that you get when you are in this regime. So, okay, so let's consider what happens when you have many types and space. Oops. Yeah. Okay, so this is one representation of one particular realization of the dynamics. So here we have a one-dimensional space, okay, one-dimensional reactor. It started with uniform concentration, and the different colors are a representation of different states of the system, okay? Because so here in general, each point of space and time correspond to a vector of concentration that you may approximate with a vector of 0 and 1, okay, with 1 indicating an active reaction with something at high concentration, and 0 representing something at low concentration. Okay, and so we are using here U-maps just to represent these differences of concentration with different colors. And in principle, the similarities of colors represent the similarity between the states. But the point that we want to make with this figure is the fact that initially you see that there are some states that persist for some time over a well-defined subset of the space. And then you have new states that appear, and some of them are able to grow. And at the end, you have one state that invade the dynamics and dominate in the reactor. Okay, so again, we have this notion of reproduction in a context also where we have a large diversity of possible states. And so this is related to this to the evolution of the productivity, okay, minus the energy. So if you look again in average in the system, you will see that the productivity increases, and you can also visualize spatially that initially you have phases of relatively low productivity, and towards the end you have more and more high productivity states. Okay, so this is like, in the language of evolution, it's like a fitness that increases with time and is indicative of what dominates over, what states dominates. If you replay the tape, if you repeat the same simulations with everything the same but the stochastic noise, then you can get many different kinds of dynamics with different states. Oops. Okay, so coming back to this slide, so this was to explain how now we have a notion of reproduction with some entities that correspond to these chemical states that can proliferate in the sense that can grow and they can replace other states. So now I want to continue and to tell you a bit about the other properties that we would like to also to observe and which correspond to things that we also see in biology, starting with selection and competition. So I've shown you that if you repeat the same evolution, we may end up with different states. Now, if we collect a large number of states that you obtain under such simulations, then we have, in fact, a large diversity of possible states, but that is different from just all the possible stable states of the system. So what this graph is showing is that if you look at the similarity between two states, and here states, you can think of it as a string of 0 and 1, with 1 being active reaction and 0 inactive, and you can just take the number of reactions that are both active or both inactive in the two states. And what this is showing is that the distribution of this similarity is very wide, both in the set of all possible states and in the set of states that you obtain at the end of these trajectories. But if you look at the productivity that is associated with this final states, again, this corresponds to the fraction of active reaction, something like minus the energy. You will see that the one that you get at the end have a distribution that is much is larger. I mean, the mean productivity is much larger than the productivity that you have for all possible random states. So this is consistent with what I said before, that in average this productivity increases, and the state that you get from this evolution are not typical states, but are selected states that have particularly high productivity. Now, what you can also do is you can do something that is similar to competition in evolutionary biology. So you take two states that you obtain at the end of two different trajectories. So this is illustrated here. And then you put them in space, one next to each other. And then you look at the dynamics and you ask what you get at the end. And in general, what you get is something that is, neither the first one or the second one, is something like an hybrid of the two. But what we can ask is whether this final state is more similar to this one or to this one. And if we do these statistics, what we observe that it's going to be more similar to the state that has higher productivity. So this is consistent with the idea that the final result of a competition is mainly dictated by the state with highest fitness, which here is highest productivity. So this corresponds to this notion of these two notions of selection of competitions. Next, we can continue and we can look for another feature that we find in evolutionary biology, which is the fact that you can have different species, or here it's going to be different states, that are specifically adapted to different environments. So what does it mean here? An environment is going to mean just what we inject in the reactor. And if you remember, there were these simple building blocks, and in particular these dimers that are dissociated to become monomers. And so previously I was considering a case where the rate at which these dimers are injected is the same for all the types. Now I'm considering the possibility that there are different influxes for different I, and a particular choice of this lambda I will define a particular environment. And so what now we can do is we can, as before, we can run a simulation in one particular environment. We can run another one in another environment. And then we do the competition as before, but we do the competition in, let's say, the first environment. And we ask whether the end product of the competition is indeed more similar to the state that evolved in the same environment. So this is what is represented here. You run a simulation in two different environments, and then you do the competition in the first environment, let's say. And what you typically get is that at the end you get something very similar to what you had here. And you can do the statistics, but the the point is to say that, okay, it reproduces this feature that you can have a specific adaptation to a particular environment, and it gives you a competitive advantage, okay, when you are in the same environment. Okay, so this is this aspect. Okay, I can. Next, you can always ask for more complex behaviors. So here we are going to talk about the possibility to have complex responses. Okay, the idea is in biology, a lot of interest is the fact that you have organisms that can respond to particular stimuli, can regulate, have regulation and so on. So here we can also look for something similar, where this time we are going to consider an environment that is changing as a function of time. Okay, so as before, the environment is defined by the influxes lambda I of these dimers. Okay, but instead of assuming that all the rates are constant in time, now we are going to consider the possibility that they change. And a simple protocol that we investigated is one in which we turn on or off one of these influx one at a time. Okay, and this way you can define sequences of environmental changes. Okay, where you decide that you start by turning on one flux, then you turn another one off, on, off, etc., etc., and it defines a particular environment. And then you can repeat something similar to what I was showing before. You have one system that evolves in a particular sequence of environment, another one in another sequence, and then you compete and you ask whether at the end you get something that is more similar to the state that evolved in the same environment. And then you can also ask, okay, to what degree you are specific to this environment. So what we have in this case is we have a form of learning. Okay, so what do we mean? So we have something that is quite specific. So what we can show is that you can really have different sequences of environment that drive the system's distinct states. And there is a notion that there is really specificity to the adaptation, in the sense that if you evolve one system in one sequence of environment, then you take this sequence and you just shuffle the order, okay, but keeping the same thing that are turned on and off. When you compete, it's really the one that has seen the environment in the right order that wins the competition. Okay, so there is a notion that you adapt specifically not only to a finite environment, but to an history of environment, and that you can discriminate different sequences of environment. There is also a notion of continuity, in the sense that if you first evolve in an environment that is similar to the one in which you do the competition, you are going to also to be more likely to win than if you were in a different sequence, where similarity can mean, for instance, the fact that you just you share the same lambda I that you turn on and off, but maybe you invert the order of one or two in the sequence. So there is a notion of specificity. So this learning is adaptive. This is what I was mentioning. You can have advantage in the competition. There is a notion of generalization, in the sense that you don't need to have seen exactly the same environment in the past to have an advantage. You need to have seen something that is sufficiently related to this past environment. There is a notion that learning is cumulative, in the sense that we can show that if you if you evolve by repeating the same sequence of environments, the more times you have seen this sequence, the better you are in adaptation. And finally, something that is interesting is that you have this learning that is not strictly equivalent to natural selection, in the sense that we can take a system we can take a well-mixed reactor, let it see the sequence of environment, okay, in condition where there is no reproduction, because there is just a single concentration at every time. and this is sufficient to reach a state that then will display some advantage when we put it in space and do the competition. Okay, so this is just an illustration to show you that we can push a bit more this kind of analysis and try to find less trivial features, something like learning of stimuli. So that's essentially what I wanted to present, this very simple model, starting from simple physical principles, but that has several features that are of interest and that are very much reminiscent of what we see in Darwinian evolution. We are not the first one to think about these kind of problems, and so our work is related and different from several previous works. So I was mentioning at the beginning, people in the context of origin of life were puzzled by this problem of how do you combine these different chemical systems. What's interesting about our model is that there is no distinction between metabolism and genetics, okay, in the sense that the same set of reactions, okay, is driving the system out of equilibrium, and then is also encoding the state of the system, okay, by having, in the sense of, so it's encoded really like digitally, okay, in the sense that you have reactions that are on, reactions that are off. So there is no question of what is first. You have the two, in a sense, at the same time. There are also a lot of efforts in the context of artificial life, where people are trying to make concrete systems that have properties that we find in biology at very different scales. One of the first proposals by Penrose was in the 50s, really using small wooden blocks. There are more advanced experiments with colloids. Also in the context of chemistry, people have also tried to make replicators. So something that is different from what we do, that I mentioned at the beginning, that in all these cases, people really focus on engineering small replicators, okay, with the idea that this is really the first thing that you need to make something that can replicate. And as I explained, in our case, there is no replication in the principles, and there is no also explicit design of a replicator. And there are some works that are a bit in this spirit, and usually they call it pre-evolutionary dynamics, in the sense there is a dynamical evolution that leads to Darwinian evolution, but initially is not itself Darwinian. And then you have also a lot of work on the more computational side. You may know, for instance, cellular automata of von Neumann. But in this case, obviously, there is no physical model, and it's not taking into account the physical constraints of the systems. I think there was another, yeah. So what's also interesting is to look at the way in which information is encoded in the system. So in most cases, starting from Schrödinger, who was talking about aperiodic crystals, the idea is that complex information is encoded into molecules, typically into long polymers, okay? And obviously DNA is a strong inspiration. There are some ideas of using what is called compositional states, which is close to what we are doing, where really the state of the system is defined by relative concentration, not by molecules. But in all these previous cases, people were really considering big molecules and assemblies of big molecules are being an important ingredient, which is not something that we need in our system. And so finally, so obviously, I mean, I was listing some features. I mean, there are many other things that we could think about and try to also have in this same system. So for instance, something that is key usually in presentation of Darwinian evolution is the concept of population. Okay, that the fact that you have discrete individuals, cells. Okay, it's not something that we have in our model, but you can ask, okay, what's other condition under which you could also get these kind of features. There is also the concept of genotype and phenotype that is very important in biology. which separates what is inherited from what is not. So it's not in our model. There is also something that is not very well defined, but that people are very fond of, which is the notion of open-ended dynamics. The fact that you continuously see new things in your dynamics. That is something that I think is also worth thinking about and trying to conceptualize. We are also interested in implementing experimentally these systems because, as I presented, we can really start from simple physical building blocks, which may be colloids in the context of source matter physics, but could also be molecules. And nowadays we have ways in which we can engineer molecules to have specific properties. Now coming back to a more theoretical perspectives, I had this table before where I was suggesting that our models combine two features that are well known. Okay, so the idea that when you have a complex network of interacting elements with frustration, okay, you can get a glassy system. So in this case, it was a chemical glass. There is this element of space, okay, reaction diffusion with stochasticity, which is also very well known. What, as far as I know, is well known is what you get when you have the combination of these two features. And this is what we have in our model. And so there is an interest in developing these aspects. The question obviously remains open whether this is really the right way to think about evolutionary dynamics, but this is the proposal that we have. And obviously there are many other theoretical questions that we may ask, but I will stop here and take your questions. Thank you.
So we have time for some questions. So maybe
Thanks for the talk. So I actually had two questions. The first one is with respect to if there is in the zero-dimensional system well-mixed case, a natural way to break detail balance. And if that is the case, where, I mean, basically you don't have this energy function that would be the productivity. So what happens in that case?
Yeah.
And I was also wondering, I guess there is a minimal size for the chemostat for this to work, right? Because if the noise is too strong, then... Yeah. So what happens when you change the size of the chemostat?
So, okay. So the first question is, I mean, we design very well the reactions so as to have this energy function. It's not something that you get typically. It's like a quasi-potential for non-equilibrium systems. And so one of the key features is to have reciprocity. So the fact that, so this is represented by this symbol. Okay, so product of one reaction inhibits the other and reciprocally. If you don't enforce this reciprocity, in general, you don't have detail balance. And I should say, I mean, several of these ideas. Okay, so I mean, you find similar behavior in the context of glassy systems, but something that is closer and was the inspiration, particularly coming from Guy, is systems of in ecological dynamics. Okay. Where you have similarly, you can modelize, let's say, Lotka-Volterra equations. And again, people have studied, okay, what are the different dynamics that you can get depending on the graph of interactions, both the, let's say, the connectivity, but also the reciprocity. And depending on this graph, in fact, you can get very completely different regimes, okay, in which you get chaotic behaviors, or you will not get diversity of states. And here the idea is really to take a system that has this potential to have this very large number of states, and then to get the energy, you need this. You also need to look at the limit of small noise. So it relates to your second question. So obviously there is also a question of having the right parameters. If you are not in the right regime, you will just not see stable states. if the noise is too large. So yeah, you need the temperature to be low enough to observe those transitions between states.
So I had one question, so related, I guess, to this, which is so how realistic? I mean, so do you expect these two assumptions that you have a very large chemical network, so I mean with many different reactants and possible reactions, and that it is symmetric, no? So that you have this.
Yeah. So here it's completely symmetric. In fact, if you have a small fraction of edges that are not symmetric, it's robust to this. Okay, so it's not it doesn't need very strong fine tuning. But then I mean, this is the question. So here the idea is you need to start from a network of chemical reactions. We don't expect every network of chemical reactions to display those features. And in a sense, this is a proposal of what kind of features are sufficient to have this behavior. And I think the question is open of if we explore more broadly the set of possible chemical reaction networks. Okay, I mean clearly some of them will not. I mean we'll have just one stable states. We don't expect to see this kind of behavior. But more generally, I mean, it's an open question to analyze what you get under different topology of chemical reaction networks.
Maybe I want just to make a comment that I think that some of these ideas that you can reinterpret this origin of life in terms of a spin glass doing aging dates back to Anderson. That I mean, he had a model of a spin glass. I mean, he had this paper on the origin of life where there were some of these ideas. I mean, he was writing a Sherrington-Kirkpatrick model for this. And the other thing is that, I mean, this is, I would expect that this is robust when you have some asymmetry in the couplings, but it's robust if the connectivity is small. So as soon as you have large connectivity, typically, I mean, what we understand now is that even a small amount of fraction of asymmetry breaks completely like some definition of approximate energy that is decreasing. So I think that this is really an important assumption.
So it's there are really two assumptions in the definition here: is sparsity and reciprocity. And regarding Anderson, I don't have all the details in mind, but I don't think that the notion of reproduction that we have here with space, which is, I think, really like I tried to explain the key new ingredients in this model. I don't think it was there in his model.
Hello, thank you for the talk. Just to continue on Pierfrancesco's question, concerning the graph, the many type graph that you introduce for those states. So you use a random graph, so it's highly connected, as Pierfrancesco said. Even though I understand that this work is supposed to be like a pre-evolutionary simulation, so it has to be like as not as general as possible, let's say. I think it's still, when you look at metabolism network, for instance, or a lot of classical chemical networks inside of a living organism, most of the time it's more small world type of networks, where some nodes have a really high connectivity, while most of nodes have low connectivity. And I think, do you think it could maybe be relevant here to try those models? And is it something you guys are thinking of?
No, I think it comes back to Jorge's question. I mean, there is a question of exploring the range of reaction networks, seeing which one have the same behavior. Maybe some of them have different behavior that is also of interest. So I think this is open. It's an interesting and open question.
Thanks for the talk. I was just curious that it's very nice to see that the models can bring you this heredity, like the parents can pass on their traits to their offsprings. I was just wondering that because in in most biological systems, we see that this heredity, like how the traits are being passed on, is usually like they use DNAs to encode these informations, and it's really that you have like specific large molecules that can encode these informations. And so I was wondering, like, do people know, or are you thinking of like extra physical principles that makes like our biological system to favor this way of passing on the information, not just that certain states are favored as time goes by?
Yes, I mean, I think you can speculate that it is more stable. You know, you can take a DNA molecule, even you leave it ambient temperature, it can maintain its information for a long time. This being said, I mean, even in current organisms, I mean, you have, you know, chemical states that are also inherited. It's not only DNA that is inherited, but when a cell divides, okay, it also transmits all the proteins and all the molecules that are in the cytoplasm. And this also contributes to heredity. So it's true that, I mean, yeah, I think in biology you have both mechanisms at work. The main dominant one that is best known and carries most extensive information is indeed DNA, but it's not the only one. But yeah, I mean, I agree. You can ask, okay, what's so in this model? I mean, there is no way in which you can make large molecules just because we chose building blocks to have interactions that are limited in one way. We could try to generalize and ask whether at some point there is a transition where information become encoded into large molecule, but maybe starting this way and later on having a different mode of information processing.
Maybe Sylvain that.
Yeah, so I have a question related to the role of energy in your models. Because in biology, it seems to me that it's not so easy to find energy, like you have sunlight or you have heat from hydrothermal vents, for example, but to capture it and to store it and to do something with it before it dissipates is not easy. And in your system, it looks like you have an easy and unlimited supply of energy with all these molecules which always come in. And isn't this related to the fact that it's quite easy to get many relatively interesting and lifelike systems? But if you had a more scarce source of energy, would it maybe restrict the possibilities to, and maybe even to something more realistic?
Okay, it's interesting. Okay, I don't have the answer. So here it's true that we provide that all the time. We provide a large collection of chemical building blocks. I think the way in which people think of the origin of life very often, this is a bit the case. So they will think of thermal vents in the ocean where you have actually a lot of chemicals that are provided. Whether you will get something very different because you add constraints on the input, I don't know. But I think I will have to think how to do it. So what we have done is we have a bit, when I was presenting the learning thing, you can ask, okay, if instead of having a constant supply of everything, okay, you start to have fluctuations, and you may have things that are not present at some point and then appear later on. So this is part of the model, the fact that you may not have a constant supply of all the building blocks. So this is something that we can explore, and how it selects some particular state that may be more robust to these fluctuations, for instance. I don't know if it's answered your question. But
Thank you.
Yeah, maybe I wanted to come back to the question mark on the phase diagram that you had. I mean, at some point you showed some pictures, so if I understand well, when you add this glass phenomenology with space, you have like some very fluctuating regions that, I mean, something that fluctuates coherently. Can you go back to, I mean...
So, you're talking about this?
Yeah, so So I mean, as far as I understand, this says that as soon as you combine the two, you can have something that is organized in space with like regions that fluctuate a lot here and fluctuate a lot there. And this is, I mean, it's reminiscent, at least to me, to what people call dynamical heterogeneities in glasses. So do you know if there is?
I don't know, but okay, I'm interested in discussing it. So something that we know that I haven't mentioned is, so here I presented it as if, okay, at the end, these are the states of highest productivity, lower energy that make it. In fact, it's not strictly the case. So the states that we have at the end are some of the highest productivity states, but it's not the only defining features of the state. And in fact, if you look at the end of the dynamics here, you have some state that takes over other ones, but they all have the same productivity. So I don't know if there is an analogy in the context of glasses, the fact that the domains that. I mean, one thing to say is, I mean, space is different, okay? Because here it's like really we have at each point in space we have a chemical. We have a system, a glassy system, okay, which is in the dimension of chemical reactions. So I think that the formal analogy is more like coupling replicas, real replicas of the glassy systems. This is, I think, formally what it is more like. So I don't know if in this context it also makes sense to talk about.
Well, I mean, if you think about some mean field model and you try to build to add space to it, so you have like some space organized model, and on each point you have a mean field model. That's how you typically build like correction to mean field theory. And correction to mean field theory gives rise to fields that fluctuate in space and that like lead to huge fluctuations. And typically what we know in glasses is that, I mean, as soon as, I mean, your system is aging, it's surfing on marginally stable states that are very sensible to perturbation, so that's somehow related to your sensitivity and learning part. And then as soon as you put it in space, it has these very huge fluctuations of mobility, for example, in glasses, and this is reminiscent to that.
Okay, thank you.
I don't want to be too long, but just to go back to that, you said that productivity in your model might not be the only variable that would select the states. But if you look at the curve, like for the sample trajectory of productivity, there are two regimes.
Yes.
That you see. So could it be that you have a first regime of competition and then a long time regime of neutral interactions dominated by fluctuations that end up in the, okay, because you have a finite system, eventually one takes over, but it's just fluctuations and there would be nothing more in that case? Or is there really additional variables that would account for the selection?
So I don't know if there are additional variables. What we know in simple models is, I mean, you can design simple networks in which you have states with equivalent productivity, but then there will one that can invade more easily the other ones, and it has to do with also the similarity of the two states. And so in these dynamics, for instance, you can see, I mean, here you have the persistence of some states for some long time. So there are states that are more difficult to mix with each other, which we can understand because they are more dissimilar. So there is an extra dimension that we partly understand here, which is the fact that when you couple your system, this coupling is also an important parameter to take into account. I mean, this is extra variable that plays a role.
So I just had one last, just very general question. So because you talk about heredity before talking about reproduction, it was a bit. So can you explain? So what it means to have heredity without reproduction?
So I mean, okay. I mean, first, I mean, it's putting words on things, and we can decide where we put the boundaries between. But in this context, it was, just a restatement of the fact that you have correlations. So if you look at successive states, okay, they are very much similar to each other. So there is heritability in the sense that you don't jump to a new state that is completely unrelated to your parents. Okay, and if you want to push the analogy, you may even say that the fact that you have aging like this is like your mutation rate that is becoming lower and lower as time goes, okay, because you tend to stay in the same state. And in space, you have the same, okay, when the things are reproduced, and you have new states that occurs, it's also related to the previous one. So this is this idea.
So I think we can stop. So thanks again. So as usual, we have some places for the launch. So if you want to join, please come. And, Thanks a lot. I think it was really interesting. Thanks.
Okay.